Today, I'm going to be speaking to you about glycolysis, gluconeogenesis, and the role of aldolase gene transcription and expression. My name is Josiah Weimer. This is Graduate Biochemistry, and here's my presentation. Alright, so to kick this off, the body is a complex unit powered by countless chemical reactions. And there are reactions that build up chemicals, and that's called anabolism. And then there are reactions that break things down, and those are known as catabolism. But some of these reactions are known as the metabolism of the body. And even though metabolism is only really associated with energy breakdown, it encompasses every single chemical process within the body. And that includes the protein production, energy production, which we're going to get into a little bit. Also, an anabolism and catabolism of sugars, which kind of goes hand in hand with the energy production. And this presentation is going to outline the pathways of glycolysis and gluconeogenesis, the way that the body builds up and breaks down sugar, the role of aldolases, which is a family of enzymes instrumental in the glycolysis and gluconeogenesis, and the different factors that influence the expression of aldolases. Alright, to kick this off, we're going to start talking about glycolysis. And this is a topic that is very commonly covered in introductory classes of chemistry and biology. But simply put, glycolysis is the catabolism of glucose into pyruvate. So it starts out as glucose molecules, and through a chain reaction of events, it will be broken down into two pyruvate molecules. And one important thing to know about glycolysis is that it's the step that initiates the process of cellular respiration. And this cellular respiration is how the majority of energy is produced in the body, so this is very important to life itself. And another important thing about glycolysis is that it provides the energy to fuel the remainder of cellular respiration. So even though it does expend some energy, it's also going to have energy to kick off the next part of the reaction, which is the citric acid, also known as the Krebs cycle. And glycolysis is going to yield a net of 2 ATP molecules per glucose molecule. And here in this diagram, you can see that glucose is broken down into pyruvate, and that releases ATP and NADH, which is it's an electron carrier, and that's just going to kick off straight into this citric acid cycle, which creates a number of products. And glycolysis itself does not require oxygen, even though it is part of the cellular respiration cycle. It can take place in both anaerobic, meaning it doesn't need oxygen, anaerobic energy production, along with aerobic production, which requires oxygen. And then glycolysis takes place in the cytosol outside of the mitochondria. So even though the mitochondria is often seen as the powerhouse of the cell, glycolysis is going to take place right outside of it. And it's a sequence of 10 smaller reactions. And we're going to dive into that part right now, and we're going to take you through it step by step. Alright, so here we are in the first step of glycolysis. We have glucose in its form, and it's a 6-member ring along with this oxygen molecule. And the first thing that's going to happen in the glycolytic pathway is that an enzyme known as hexokinase is going to add a phosphate group onto the glucose molecule, and you can see that right here. And in this process, there's one ATP that is expended. It's dephosphorylated, and that releases some energy. And that phosphate group is actually going to go straight right here. And that's going to lead to a molecule known as glucose 6-phosphate. And it's basically the same molecule. All that's different is that it has a phosphate right here. And the next step is known as catalyzed by an enzyme known as phosphoglucose isomerase. And whenever you see the word isomerase, that basically means that the molecule is changing form. That's the main takeaway for phosphoglucose isomerase. It's going to change glucose 6-phosphate into another sugar known as fructose 6-phosphate. And that's going to set us up for the next reaction. And this next reaction is phosphofructokinase catalyzed. So what this one is going to do is it's going to add another phosphate group right here. And you can see whenever ATP molecules are used up right here, it is often associated with the phosphate group being added. So whenever that phosphate group is broken off right here, this enzyme is going to grab that group and put it on right here in place of this H. So this H is ripped off and that phosphate group is placed on in its stead. Alright, and here's where it gets a little more complicated. This is actually where the aldolase kicks in, and we're going to actually get into a little more depth for the aldolase in just a little bit. But in this step, the aldolase is actually going to cleave between the C3 and the C4 bond, and it's just going to break this fructose kind of down the middle, and you're going to come out with two molecules. We're going to have one dihydroxyacetone phosphate, and then we're going to have a glyceraldehyde 3-phosphate. You can see that right here. And even though these are two different molecules, there's going to be another enzyme that kind of kicks in, and this is known as the triose phosphate isomerase. And this is just basically going to rearrange this molecule, the dihydroxyacetone phosphate, back into the glyceraldehyde phosphate. And whenever you see isomerase, that means that the structure of the molecule is going to be changing. All right, and whenever we have the two glyceraldehyde 3-phosphates, that kicks us into our next step, which is glyceraldehyde phosphate dehydrogenase. And well, that's the enzyme, the glyceraldehyde 3-phosphate dehydrogenase. And it's going to form a molecule known as 1,3-biphosphoglycerate. And this is just basically adding a ester group onto this molecule right here. And in this process, NAD plus becomes NADH. And this NADH is an electron carrier, which is going to play a key role in oxidative phosphorylation in the electron transport chain, which is where the majority of the ATP is made. And this 3-phosphoglycerate is going to become 2-phosphoglycerate, and this is through the process of an enzyme known as phosphoglyceromutase. And basically in this one, the phosphate group is going to move from the third carbon up to the second carbon. Alright, and the next step is the 2-phosphoglycerate becomes phosphoenol pyruvate. And this is kind of our second to last step. And we have an enzyme known as enolase. And this enolase, what it's going to do is it's going to take off this water molecule. And that water molecule is going to just get right out of there. And it's going to form this compound known as phosphoenol pyruvate. And as you can see on this phosphoenol pyruvate, it's almost to the point of being pyruvate. All we have to do is remove the phosphate group, and that's where the pyruvate kinase kicks in. And in this step, an ATP molecule is also made. And you have to remember that since we have two Since we have two of these molecules becoming pyruvate, that means two ATP molecules are going to be made here and here. So glycolysis is an energetically favorable reaction because it produces more ATP than it expends. And that kind of sets the stage for the rest of cellular respiration to take place. All right, and you can see right here that the pyruvate molecule is made, and it's ready to be turned into acetyl-CoA in the next reaction, which is in the Krebs cycle. But that's another talk for another day. All right, the next pathway we're going to talk about, it's equally important. It's known as gluconeogenesis. And kind of like the name suggests, it's responsible for the production of glucose within the body. And it can convert all kinds of different substrates into glucose. And these substrates can include lactate molecules, amino acids, and glycerol molecules. And you're going to see that whenever I walk you through the gluconeogenesis pathway as well. And another important thing to note about gluconeogenesis is that it takes place when blood glucose is a lot lower. And one example that I like to use is ketogenic dieting. And you've probably heard of the fads with ketogenic dieting where people are eating as few carbs as possible in order to burn up fat in their body more effectively. And instead, what the body is going to do is it's going to take other substrates, including chemicals with ketones in them, and it's going to turn them into its own glucose. So since we're not giving our body any glucose when we go in a ketogenic diet, what's going to happen is our body is going to take what we have been eating and it's going to make glucose of its own. And another thing important to note about this is that the gluconeogenetic pathway is inherently endergonic, which means that it's not energetically favorable. But whenever it's coupled with ATP or GTP, the process does become exergonic. And I'm going to demonstrate this in our next slide. Alrighty, so gluconeogenesis, similar to glycolysis, has a number of different steps to it. And the first step of gluconeogenesis is the conversion of pyruvate to phosphoenolpyruvate. So we're basically working from the bottom of glycolysis all the way back up to glucose. So what's going to happen whenever pyruvate is turned into phosphoenolpyruvate is that we have this enzyme known as pyruvate carboxylase. And what that's going to do is it's going to convert the pyruvate to oxaloacetate. And that's just a kind of an intermediate right here. And you can see here that ATP and CO2 are also going into the reaction. So something that was kind of not energetically favorable, whenever we add ATP into that, it makes it much more energetically favorable. And we have the release of that. And since we have a release of a phosphate group right here, what's going to happen is it's going to be added on to the pyruvate. And that's going to give us the phosphoenolpyruvate molecule. And I can show you right here. We got that phosphate group again. And that it didn't go anywhere. All it did was it bound to this ATP. And that's how ATP was produced. But in this reaction, it's backwards. So the ATP is burned up in this part. And it forms phosphoenolpyruvate. And then the second step is we have the phosphoenolpyruvate carboxykinase, or PEP-CK. And what that's going to do is it's just going to finalize that transformation from oxaloacetate into the phosphoenolpyruvate. Alright, and then another important thing to know is the steps 2 through 6 of gluconeogenesis. We're just taking the phosphoenolpyruvate and we're going to turn it back into fructose 1,6-biphosphate. And this is essentially a reversal of the glycolytic process. So that means that there really aren't any new enzymes or anything like that that are taking place. It's just working in reverse. And a lot of these reactions, as you're going to see, are reversible, which you can see by these double-headed arrows. So we have the phosphoenol pyruvate turning back to the 2-phosphoglycerate, and that means you're going to have a hydrolytic reaction instead of a dehydration reaction, because we're going to introduce the water back into there. So we're going to have, what's going to happen is this molecule right here, we're going to introduce the water back in, and it's going to add a water back there and a hydroxyl group right there, and it's going to turn into this 2-phosphoglycerate. Alright, and then after that we're going to have phosphoglycerate mutase, and it's going in reverse again. So what's going to happen is that 2-phosphoglycerate is going to move back up to the 3-phosphoglycerate. So the second carbon is going to move back down to this third carbon. And then right here, we're going to have another release of ATP to ADP. And this is just making the process energetically favorable again. But since this is happening, since we're having an ATP burned into an ADP, what's going to happen is we're going to have another phosphate group that's added on. And you can see that right here, the 1,3-biphosphoglycerate. And then the next step is we have the glyceraldehyde 3-phosphate dehydrogenase. In this process, you're going to see these NADH drop off the electrons, and it's going to be oxidized back into NAG+. And that's just going to make it a little more energetically favorable as well. And then we have this next step. And this is the step where we divided it all into two different molecules. We have the glyceraldehyde 3-phosphate, and we have dihydroxyacetone phosphate. And what's going to happen is instead of breaking a fructose apart, in this part of the reaction, aldolase is actually going to reform that. And this is important to the process of gluconeogenesis, since the body is responsible for making its own sugar sources. Okay, and then in this reaction here, this is where it stops being simply a reversal of glycolysis, and it's going to have a different kind of reaction. So in this one, the fructose 1,6-biphosphate is going to be dephosphorylated to fructose 6-phosphate. And this is going to be done by an enzyme known as fructose 1,6-phosphatase. You can see that in this diagram right here. This puts it back up to fructose 6-phosphate. And then this next step, phosphoglucose isomerase, it's just going to change it back into glucose because isomerases do change the structure. That's what they're known for. And then the last step is glucose 6-phosphatase, and what that's going to do is it's just going to yank the phosphate group right here back off, and that's going to form your glucose molecule as we know it. Alright, that brings us to our next concept, which is the role that aldolase has in glycolysis and gluconeogenesis. And aldolase is important because it catalyzes the conversion of fructose 1,6-biphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And you can see that in this diagram right here. We have the fructose 1,6-biphosphate, and we talked about this in the other pathway, but it's just a good reminder that aldolase is going to break the bond right here, and that's our 3,4 bond. It's a little easier to see whenever it's in linear form. But it's going to form the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And what it's going to do is it's going to form what's known as a Schiff base with a lysine residue. Now this lysine residue is found in the active site of the aldolase, and is responsible for helping ultimately break that bond. And these two molecules are going to undergo that series of reactions that form pyruvate. And then, kind of like we were mentioning, in gluconeogenesis, the aldolase is actually going to unite those two glyceraldehyde 3 phosphates and dihydroxyacetone phosphate back into the fructose 6 biphosphate form. Alright, the next part is isoforms of aldolase. And there are three forms of aldolase that are involved in metabolism and glycolysis in the human body. And the first one is aldolase A, or aldol A. And this aldolase is highly represented in fetal tissues. And in adults, it is mainly found in muscles and red blood cells. So it isn't as common as aldol B. And it's also found in the cytoplasm of the cells. Aldol B is the most significant one you're going to find in the human body. And this one is the one that's instrumental in fructose metabolism. And it's most heavily found within your liver, kidney, and small intestine. Aldolase B is stimulated by glucose and insulin. So that basically just that's important because aldolase is very instrumental to glycolysis. And whenever the level of sugar in your blood is high, your body is going to release insulin, and it's also going to try to break down that sugar into usable energy. And another important thing to note is that aldolase is inhibited by second messengers such as cyclic AMP or CAMP. And then that brings us to our third form of aldolase, which is aldolase C. And that's found in the brain and nervous system. And it's not as common as the aldolase A and B. And it also has higher areas of electronegativity. And those are subject to a lot of different scientific research at the time. And a lot of scientists believe that it does have a potential role in cancer, since cancer cells do tend to use glycolysis very liberally in what's known as the Warburg effect. One of the main ways that cancer cells produce energy is through the use of cellular respiration. Alright, and that brings us to our final point, and that is the transcription factors that influence aldolase production. And kind of like we were saying in the last slide, aldolase B is the most highly expressed in the liver and kidneys, where it's instrumental in breaking down fructose and also the formation of glucose. And there are multiple factors that help promote or inhibit the expression of aldolase B based on what the body needs. One of the most important ones is HNF1, and that one is a strong activator of the aldolase B promoter. So what it's going to do is it's going to bind to a promoter, and that's going to be represented here by this active site. And it's going to bind here to this promoter and allow it's going to allow the production of this aldolase B protein. And then HNF3 is significant because it is an anti-activator of the aldolase B promoter. And what it's going to do is it's going to competitively inhibit the activation of HNF1 and by extension the activation of aldolase. And whenever we say that something is a competitive inhibitor, it means that it binds in the exact same spot. So as you can see right here, here's our substrate with enzyme A. But in here we have something known as a competitive inhibitor. It's binding directly where this would have. So that means it can no longer bind there. Another thing about HNF3 is that it non-competitively inhibits the activation of DBP. And DBP is another factor that will help activate and promote aldolase B production. And that one is a non-competitive. So that means it binds to a different place than where the DBP would bind. But since it bound there, it's actually going to change the structure of the enzyme in something known as an allosteric inhibition. And it's also known as non-competitive, since it's not taking up the spot, but it's still inhibiting. All right, and then we have the CEPBs, and these are families of proteins that form dimers with other factors, and this can include DBP. And within in vivo samples, this turned out to be a weaker activator of the aldolase B promoter, but in isolated cells, it did show a much stronger representation, and it's also going to transactivate in the absence of DE factors, which kind of stop the promotion of aldolase B. Our last one is DBP, which are D-element binding proteins. And these are strong activators of the aldolase B promoters. So DBPs and HNF1s often do work hand in hand in order to allow aldolase B enzymes to be formed in the liver. And that brings us to the end of our presentation. And a little note that I'd like to leave you with is that our bodies are a masterful design of a great creator who knew what he was doing when he breathed us into existence. And he's given us a mind to understand the intricacies of the body. So the question I want to leave you with is, how do we use this gift that he's given us? Thank you and have a great night.